Okay, wait.
sir, अभी कॉल कैंसिल होना है
You decided to stay, right? Yes, we can start?
Yeah.
Okay. So let us start. So this is the end of the torture. This is the last lecture. We're talking about stochasticity, randomness in chemical biological processes, and we're doing this by examples, learning by examples. I hope you learned something. So this is about precise timing in biological processes and the story of bacterial cell lysis. Again, I already showed this picture. There is an expected precision in biological processes. If you remember, this is a plan. One is far away. Okay. Anyway, this is a plant which grows in some desert. In what country? Yeah, there's a desert, but what country? Two countries. Yeah, I mentioned two countries. What two countries were sharing this desert, remember? No, Peru and Chile. So this is a plant which grows in this desert, which is on the border between Peru and Chile. Beautiful things. It grows exactly 1.5 centimeters with a precision up to millimeter, which is always amazing. Okay, I have to go with on. Another example of precision is cell division, as we mentioned. Despite the fact that it's quite complicated, the typical once per 24 hours. It's very, very precise things. We also went over this example. So remember P53 proteins, which is indicator of stress. It increases until it reaches some threshold and bam, cell dies in the process known as apoptosis. We also argued why this is strange, right? Why, or at least why this is not so easily to explain, because all biology is a lot of chemical reactions, and if we get one chemical reaction to another chemical reaction, then you naively expect that the process which combines those two chemical reactions will have a broader distribution of reaction times, but that's not the case. So there is a very interesting example, another example of precise timing, and that's bacteriophages. Bacteriophages. So bacteriophages are viruses which affect only bacteria, right? So the way they work is they go into bacteria, do something, hijack the genome, and then they break the bacteria, and many more species appears, and then they go away. Cell lysis. If you look at a little bit more details, what's going on here, it's so the virus inserts parts of its own genome into the genome of bacteria and stimulates bacteria to stimulate the production of proteins. One type of protein is it produces a whole foreign protein, come from a whole foreign proteins, and they actually this protein then go into membrane of the bacteria. They start to increase, and then suddenly, when they reach some number, some threshold number, they create holes, and this is the end of bacteria because lysis happens. Bacteria just breaks, explodes. Then the story is a little bit more complicated. If you probably know, some bacteria has two membranes. There is another protein is produced, but eventually this is one of the most important events. And the protein called Holins because it creates holes. Okay? Very original. Good. So the reason I'm talking about cell lysis because it was quite well measured. There is a significant number of experiments, and quantitative measurements is good enough for us to understand what is going on. So the main player here is a protein called this one actually, no wrong one, this one. This is protein which is called colin. It has three domains which go into membranes here, then eventually inside of this membrane they aggregate, create holes, and that's what kills the bacteria. That's what kills the bacteria. So there were a lot of experiments was done. People were really able to measure quite precisely the lysis time before bacteria is killed. You can see some of these measurements. And what was also nicely done here, in a typical biological way, one of the most important biological ways of studying something is mutation. They do mutation in one places to prove the new species is similar to what you had before, then repeat the measure. Okay, so it's very convenient. It's similar to perturbations in physics or perturbations in quantum mechanics. That's what they do.
So I'm sorry, lysis versus apoptosis. Apoptosis, you said is cell death.
Yeah, so that's
Lysis is
it is a death.
the
It's
same thing.
just the same thing. Apoptosis typically happens through internal mechanism, and lysis is really breaking the membrane by force. Okay, so when they did the measurement, there is some interesting observation came out. So this is a measurement, again, mean first passage time on lysis time before bacteria disintegrate, and this is a coefficient of variance, which is a measure of noise. And this was done for 30, I think it was 21 mutants. 21 mutants. And there was an interesting observation. So first of all, they thought at the beginning that the wild type species bacteria are the most efficient, kills bacteria the fast. And it's not. There were some mutants which could do things faster. See, this is a mutant. This is a wild type. But the wild type produces the smallest possible noise. Smallest possible noise, you see, it's a minimum. It's the smallest possible noise. The question where it comes from. So there is even for wild types, there is a significant distribution of time before lysis. Wild type, this is one of the mutants. This notation means that the residue number 87 is changed from I to L, and this is from 89 from S to W. It's a label for amino acids. We don't need to know it's changes. Okay, good. So this is a typical experimental measurement for what they see. And clearly, this is again an example which is very nicely can be described by first passage approach. And the problems was that people still cannot understand what is the microscopic origin of this threshold. Okay, and why there is a threshold. So in other words, the scoring proteins increase, the number of coding proteins in membrane increases, nothing happens, and then suddenly, boom, when it reaches some threshold, it breaks the membrane. Why? What is going on? We needed more molecular view. We wanted to understand what is these properties of foreign proteins also govern the ability to. And again, when it comes to stochastic hypothesis, right? Stochastic model, as you have seen 9 times, you can do some stochastic hypothesis, and it really can explain many phenomena. So this is a model. This is an idea. The idea, as I told you, is that you can come out with almost deterministic outcome if you properly couple some stochastic process. Remember, I was already had some similar approach when we talked about cell size regulation, right? If you properly couple stochastic process, you can come out with an almost deterministic approach. It's the same hypothesis, but let me show you that it does work here also quite well. Again, the model very similar. You come out with a very similar model. You are again combining only 2 stochastic processes. What are these processes? These are so these numbers correspond to state of the system. how many foreign proteins are in membrane, 3 foreign proteins in membrane. That you're assuming that the foreigns are entering from the cytoplasm. So proteins are produced in cytoplasm, then they enter to membrane. And you assume they enter with the same rate A. But then lysis can happen with rate constant R. And you assume that lysis, the more foreign proteins inside the membrane, the more probable is a lysis. Extremely simple, one-dimensional model, two perpendicular phenomena. One stochastic in this direction, in this phase, in perpendicular lysis. This is a model which, so this is a model, and the assumptions are the following: that the accumulation rate, which combines several processes, diffusion, entering membrane, spreading, but you can lump everything into one process of entering membrane. There is a lysis rate is proportional to number of points.
I think you
Too close to the case. It's proportional to the number of foreign proteins inside, and you assume there is no protein degradation. So experiments show that for this protein degradation is very slow, so you can neglect it. So remember, when we do the model is named to theoretical people. It's a caricature of real world. It means that you throw away something which you think is not important, you keep something that is important. So I throw away, there is a degradation, but it's very slow. But for my model, I can throw it away, okay? Good. This is stochastic model. Clearly, the way it's described, you want to do it with the first passage. You don't have to. As I argued, you can do also with the normal master equations, so-called forward master equations, or you can use it from first passage. Again, solutions, you can do it. And again, it's similar to models that we use for cell lysis, but there are differences, of course. There are differences because the probability of lysis is actually position dependent. But the important quantity is this one. This is the quantity, the probability for cell lysis to occur exactly in state n. The physical meaning of this quantity is very simple. There is an n-1 event unsuccessful lysis, event number n successful lysis, right? And why I'm trying to calculate it? I'm trying to calculate it using experimental numbers. That's because I'm getting something like a threshold. See, I'm getting the peak. That's what I'm associating with threshold. But the most important thing is I don't need to assume threshold. Threshold comes naturally from stochastic model. So that's why we need this picture. And the role of important parameter played by this coupling, which is the ratio of entrance rate over lysis rate. This is this quantity x. And you can see you can associate it with the threshold. Because strictly speaking, in biology, the threshold is not exactly, let's say, 1,000 proteins. The threshold is also a little fluctuate, similar to what is observed. Good. So this is the most important result. I can continue with mathematics, but the most important result, this miracle of thresholds, you can explain it. And you can explain it extremely deterministic concept of threshold with two stochastic random processes. Two stochastic random processes which work perpendicular to each other can produce behavior like threshold. Okay? And if you remember, in cell size regulation, they were able to produce very precise sizes for the same reason. Okay? Good. So, but we can do more. So we in this case, the model is relatively simple. We can really calculate first passage distributions. And these distributions are, in principle, should describe the distribution of the lysis time. So we decided to fit this experimental data with one fitting parameter, essentially one fitting parameter. We were able to fit it for 21 newtons. We did it, all of this, and we collected the data, the result of the fitting, and the data are over here. So let me walk you through. So the first row is wild types, and then there is and then all other are mutants. This is the labeling comes from this experimental work. I don't understand what the label is great. Mutants, we don't need to care. We calculated this effective rate of entrance into membrane. We fit it. We estimated the effective lysis rate. We estimated this coupling, which is the ratio of entrance rate over lysis. And we also estimated this maximal number of colonies at which lysis happens. process. So, and it's always interesting to compare those things here, to look over. And that's what you can see. You can see something interesting. You can see that, so first of all, there is there are two orders of change in the entrance rate. You go for all of them. There is only one order of change for lysis rate. Coupling is also two orders of magnitude. But the most interesting thing here is that our theoretical approach predicts the number of Hollings which is consistent with the experiment, and number of the Hollings for wild type at which lysis occurs is the largest. You understand? This is the largest number. If you look at Montefiore, it's the largest possible number. So the wild types are somehow wild type species wants to have as large number of foreign proteins as possible. So this is the result of our fit. So this is noise. This is the noise. This is average number of foreign, and you can see that wild type is always the largest one. These are all the different mutants. Wild type produces the smallest possible noise. And you can see the theory experiments agree well. Theory is red one, experiment is blue one. So it seems one of the first conclusions is that if you increase the number of proteins in membranes, the noise will be lower. And this is expected, right? We know this. So the more things, the more people, then the relative measure of fluctuations is less. Increasing the number of species decreases overall the noise. And the smallest noise is observed for wild type species. But interestingly enough, it's not the fastest. So wild type bacteria are not the fastest. Again, we come to this picture that I showed you. Remember, it's not the first time we had this dilemma. Speed selectivity trade-off, typical in biological processes. If you want to do something faster, will be not precise. If you want to do something precise, will be slower. Remember, we encountered this problem in biological error correction, right? But in biological error correction, if you remember, nature chose what? Speed or precision? Speed. Nature chose speed. Here, nature chose precision, not speed. Right? You understand? So nature really is full of, I don't know, many tricks, many things. It depends on what is needed. Nature can choose different things. So it's not always speed. For error correction, I was arguing why it's speed, right? Remember, you probably remember on the Chinese car factory.
Yes.
As long as it's tolerable quality, right, a low salary, that's how it works. That's the economical reason. I don't know why the reason is here precision. I think I know a little bit. I can speculate later. But I want to tell you that biology is very versatile. There are many possibilities you can do, and nature can choose things what you want. Again, all of this comes from stochastic analysis, which allows you to compare things and to look into this problem from a different point of view. Okay, this is my speculation. This is my speculation why in this case nature chose precision. So what is the goal of virus? What virus wants? Tell me, what is the reason for virus? What is the main goal for virus when it infects bacteria? What do you think? But how, so okay, let's wild virus goes into bacteria. How many viruses you need to come out after you break the bacteria? You put money in the bank, you get a little bit more with the percentage, right? It's much more here. You play in stock market. You want because you want more money. You don't want to get the same amount of money you put in. Virus wants to do the same thing. It goes into bacteria, hoping that after bacteria is broken, more species come out. Right? If this is the case, again, this is my speculation. If this is the case, this lysis time cannot be too short. Cannot be too short, right? Because you do not have enough time. You need to create a new species. But it also cannot be too long. Why? Because evolution is a constant war between bacteria and viruses. Bacteria is always looking for sources to kill virus, to eliminate it. And the longer virus stays in bacteria, the worse for virus, because bacteria will find a way. Which means there is some optimal time, and you need to have some precision. You understand, right? That's my speculation. So the reasoning here is different from error correction. You need to be precise, more or less precise, be very narrow range of of times. Okay, so analogy that I give to people who take chemistry, chain reactions. You know chain reactions, right?
Yeah.
Atomic explosion, whatever, chain reaction, burning of something. When you molecules react, you create more molecules, and create more molecules, it's like explosion. So that's what viruses, they kind of program this way. They need to always increase, okay? Good, the virus must produce. So we look over at this data again, and we notice the following thing. So if I look at this is a wild type. If I look at wild type, or I look this way, this average number of foreign proteins, the wild type is really special case. The wild type systems are not only the most precise, they also have a narrow distribution of proteins inside. So it seems that a wild type system is able to accumulate the largest number of proteins, and it also characterized by the narrow spatial distribution. So wild types are probably optimized to do these things by doing two things. To insert into membrane the largest possible number of proteins and somehow maintain the relatively narrow spatial distribution. That allows to have the lowest possible noise and to work the most efficiently. This comes from the stochastic analysis, and you can clearly see here, okay? This is a wild type, and these are all different mutants. Okay, so you can see that all these mutants, they cannot satisfy these both conditions. Or they fast enough, and then they vary, or they fast enough, but the distribution is high, or their distribution is low, but they are too slow or too fast, okay? So this is a molecular picture. So the polyproteins inside membrane aggregate to create holes. Then, but you can ask another question, what is the mechanism, why they aggregate, why these proteins aggregate, what is the most relevant properties here? And here, this is the structure of this protein, written in the language of amino acids here, and you can put it in AlphaFold. Everybody knows what is AlphaFold. Everybody knows what is AlphaFold. What was Nobel Prize this year awarded for what? In chemistry. AlphaFold, essentially, for people who come out with the AlphaFold. AlphaFold is an artificial intelligence machine learning tool which allows you to predict static structure of all proteins. Okay, so you don't need to do experiments now. AlphaFold more or less can give you things well enough. Okay, so our idea that we wanted to do, we wanted to understand what chemical or physical properties of these proteins are responsible for the ability to aggregate and make holes. And we did the analysis. So we looked, there is the database from which you put the chemical formula and give you the properties, and then you can find the correlation between many properties and outcome. And then you do Pearson correlation coefficient, and you can determine the most anti-correlated and the most correlated things here. And we were able to discover several of the properties. But for this talk, I only look at one of them, hydrophobicity. There is hydrophobicity. There is a clear correlation between hydrophobicity and lysis time. If something is more hydrophobic, it will have less time to break the membrane. And the reason is the environment inside of membrane is hydrophobic. So many proteins will come together. The hydrophobicity increases the probability of them to come together and to break their system. And this shown over here is all different mutants here, and you can see we did this figure analysis, so statistically relevant. and the analysis is, this is my explanation. That's because inside of membrane is a hydrophobic environment. So if you connect several hydrophobic things together, you can break the membrane because you are substituting the piece of membrane with your aggregate, which has the same hydrophobic property. So now the correlation was so strong that we could predict something. We could say, suppose if on 21st amino acid, we can do different mutations, and that were our predictions for times, and this is for hydrophobicity. So the value of theory, I think I told you this, not that it can explain something, the value of theory that it can predict something quantitatively that can be tested. And this is a prediction. This is a prediction. You can do the same here for so we show this for 3 different locations, 21, 38, 85, where you change one amino acid to another one. Okay, so as I told you, this will be relatively short talk. So this is the story. This is the end of the story. Oh, I made a mistake. Stochastic framework can explain many things. An example, the reason I gave this example was to show that it's not always speed. Sometimes precision is better in biological system, and you still can get all of this through stochastic model, right? For stochastic model, stochastic framework, you can analyze the data. It's powerful enough to do a lot of things. So, and we also show this kind of obesity is critical for efficient action of point protein. Now, we did a little bit machine learning here, but it's not enough to talk about it. Okay? So this is the end of my lecture. And before you ask questions, I want to thank you for your attention, for asking questions, for being there. So you have to remember, it's talking to young people, you already established. You will someday you will be teacher or whatever, scientist, researcher. You will need to communicate with someone. This is the most difficult thing, not even in science, even in your family, whatever. The most difficult thing is to communicate with other people. So it was difficult for me. I was trying to do it. Learn how to communicate, and you will succeed in your life. Thank you so much. Questions. Now you can ask questions if you wish.
Does the mathematical model again come down to some lattice and
Yeah, it is again lattice model. It's again lattice model. Very similar to what I did, to what we discussed with you at,
there you go.
this is. Again, this is an example that by the same mathematics you can really look at different phenomena. The outcome is different. Outcome is different, in my opinion. Interesting, not the same that it was, for example, for cell size regulation. But that's we can do it.
Any other question?
This is the last chance you have to ask any question.
No, let's thanks once again the speaker.
Did we give certificates or we give them?
Close the session.